Hello, everyone, and welcome to today's first lightning talk session. We're session number 5 on the theme Biology and Conservation of Tropical Birds. My name is Sarah DeWalt. I'm professor and chair of the Department of Biological Sciences at Clemson University, and I'm also the past president for the ATBC. And today I'm going to be moderating our session. We have 10 talks, 10 lightning talks, each 3 minutes each. And what we will be doing is starting a video soon that has all 10 talks, one after another. And I'll present the speakers in just a minute. If you have questions, and I hope you do, please put them into the Q&A here in Zoom. So down at the bottom of your Zoom session, there's a little Q&A. Go to it and then write the name of the speaker to whom you would like to ask a question, and then put your question in. And at the end of the videos, at the end of all of the talks, I will pose the questions that are in the Q&A to each of our speakers. And we can have a great discussion about tropical birds afterwards. We'll have about 40 minutes for questions and answers. So we have a nice amount of time to discuss after the sessions. So today's speakers we have coming up. First we have Vitek. Oops, I just lost it. First we have Vitek from LSU, and then Akshay Surendra, and then we have, let's see, from Yale University, and then we have Mauricio Rodriguez Bardia from the University of Costa Rica. Then Paula Francis de Oliveira from the University of Brasilia, Laura Bizzarri from the University of Connecticut, Stephen Sevillano Rios from Cornell University, and Tede, Adriana Bravo from the University of Toronto, Supung Galapathi from Guangxi University, and then Luisa Gaia de Souza from the University of Brasilia. All right, so with that, Zach Zahawi is our Zoom technician today, and he's going to be starting our webinar. So again, please put questions at any time into the Q&A, and at the end of the session, we'll be able to address those questions. All right, thank you. Go ahead, Zach.
Thank you for tuning in. My name is Vitek. I'm at Louisiana State University, and I'll talk to you today about light and thermal niches of Amazonian birds. Our study site is the Biological Dynamics of Forest Fragments project located in central Amazonia. That project was established now about 4 years ago, and it was designed to study the effects of forest fragmentation on biodiversity. And what we've learned is that, at least for birds, being in a fragment is generally not a great thing, especially if you're a member of terrestrial insectivores. This is an ecological foraging guild that includes species like leaf tossers, antpitas, and birds. So these species are sensitive to forest fragmentation. And we've learned it about 20 years ago now. What we've done recently is also to look at what happened to species within intact areas outside of fragments. And we were surprised to learn that even out there, again, away from fragmentation, these terrestrial insectivores have declined in abundance. And the question, of course, is then why? Why is this happening? And to start chipping away at that, we can go back to fragments. And there, one of our proposed mechanisms for decline was tied to microclimate, because these fragments get brighter, they get hotter, they get drier because of edge effects. And those conditions may be novel and unsuitable for these forest species. But if that's the case, if those same conditions are also changing within primary forests because of climate change, perhaps habitat quality in primary forest is getting lower and lower. To start looking at this, we first want to ask the question, do they really prefer dark and cool microclimates as we assume they do? And so for that, our objective was to quantify light and thermal niches of these species. To do that, we took catchbirds with mist nets. We placed loggers on their backs, and those loggers then collected light and thermal data. We managed to catch 71 individuals of 10 species. We recovered 33 of them at least a year later. We also placed the same sensors in the environment, and altogether we collected about 5 million data points. And this is what we found. This is the light curve over the course of the day in an open area. So this is a forest gap, or this is a pasture, and we are measuring the light intensity. The light intensity, though, clearly is lower on the forest floor where these birds live, and in fact it's about 2.5% than in the open area. But the birds, surprisingly, have used even darker places than on the forest floor, than the average conditions on the forest floor. And in fact, the open areas, the light intensity there is about 2,200 times stronger than what the birds experience on average. So that's light. Let's look at temperature. I'm showing the species-specific thermal curves as measured by the loggers, and you'll see that when compared to the air temperature on the forest floor, it is much higher. And it's clearly because birds themselves produce body heat. They're endothermic. So to try to disentangle those two things, we also measured body temperatures of these birds. And then we look at the difference between the mean body temperature and the logger temperature to create what we call the thermal margin, which is essentially just a measure of the bird's thermal environment with respect to their body temperature. So you have species like this Spermathera are living pretty close to its body temperature, and there are species like this Sclerurus that live relatively far from their body temperature. So do they really prefer dark and cool microclimates? I think we have good evidence to suggest that they do track low light conditions on the shady forest floor. It is much more difficult to make an assessment about their thermal niches because of that link to body heat. But they're clearly not selecting hot areas. And I think the important thing is that climate change is working against those conditions that appear to be preferred by these birds, and perhaps maybe the reason for their decline. And with that, I'll leave you with these acknowledgements, and thank you for tuning in.
Hello everybody, I'm Akshay Surendra and I'll be talking about bird community dynamics in rainforest fragments, insights from 2 decades of bird surveys in southern India. Forest loss and fragmentation is the new normal. About 70% of all remaining forests on Earth are less than a kilometer from the forest edge. Future forests are likely to consist of smaller rainforest fragments and many more of them. Many protected areas themselves are isolated forest fragments, making it more important than ever before to understand whether diversity in these forest fragments are retained over time. The static picture of diversity in forest fragments is well known. For instance, we know that large connected forest fragments are better than small isolated forest fragments. Habitat quality matters, as does the quality of the matrix. But what about changes in diversity over time? Island biogeography theory predicts that species diversity in forest fragments will eventually reach a dynamic equilibrium. In other words, colonization rates will match up with extinction rates. However, long-term studies that have tracked forest fragments since the event of fragmentation find transient dynamics driven by factors such as extinction debt persisting for over 3 decades. So steady-state dynamics remains poorly understood because it occurs much later. So the question we are trying to ask in this study is, do forest fragments that have reached steady state dynamics presumably retain their biodiversity over time? Or in other words, are fragments of today worth protecting tomorrow? We asked this question using birds as a focal taxa. Here's what we did. We used fixed radius point count sampling to study 19 forest fragments in and around the Valparai Plateau in the Anamalai Hills in the southern western Ghats of peninsular India. We sampled birds over 6 years. In the first 4 years, T.R. Shankar Raman, a co-author on the study, collected the data. Data for 2005 was collected by Hari Sridhar and 2019 by myself. In total, we had 2,223 point counts, averaging at about 340 point counts every year and about 40 point counts every year in every fragment. All the birds we detected were passed into two categories, either rainforest birds or open country birds, based on previous studies from the region. We used three response metrics: species richness, where we controlled for the number of individuals; density, where we accounted for imperfect detection; and compositional dissimilarity, which was calculated at a fragment level across time. We used three kinds of predictors: spatial predictors, habitat quality metrics, and time. We used a linear model with a multiple regression framework to estimate our responses and relationships. Coming to our predictions, in this talk, I'm only going to be talking about the marginal effects of time on response. In other words, all other factors, predictors held constant. In the subsequent figures, you will see subplots as follows. The first subplot will be richness, the second will be density, and the third will be compositional dissimilarity, all of which will be plotted against time. Remember, these are the marginal effects of these responses. We suspect three scenarios would happen. These metrics could decline, increase, or stay constant over time. We predict that under scenario where fragments are worth protecting, species richness, density, and composition will remain fairly constant over time. However, on the other hand, if the community is unstable, declines in species richness and density over time, then fragments are in trouble. Remember that the fragments that we are currently studying in the Valparai Plateau have been isolated for the past 70 years at least and remain, we suspect, in steady state conditions. Here are the results. The species richness of open country birds declined over time, while the density remained fairly constant. Communities further apart in time were less similar to each other. Rainforest birds, which comprise most of the bird community in these forests, declined in species richness over time, declined in their density over time, and increased in their compositional dissimilarity over time, much like open country birds. It's clear that the diversity of rainforest birds is declining, yet the decline in richness is small at about 6%, while the decline in density is substantial at about 30%. In other words, we're getting similarly speciose but sparser bird communities over the last 2 decades. This has serious consequences for ecosystem functioning, especially the decline in the density of birds. These declines are likely driven by regional and global factors because the fragments themselves are relatively stable. In fact, some of them have been restored. These causes are large scale and therefore likely to persist, but will the effect on diversity continue in the future? It remains to be seen. Yet these fragments are worth protecting because they retain about 94% of the richness even after the last 2 decades where densities have declined. The references, and we have a number of people to thank. Thank you for listening.
Hello, my name is Mauricio Rodriguez from Escuela de Biología at Universidad de Costa Rica, and I will present our research on genetic structure in birds with different levels of tolerance to urbanization, the case of the white-eared ground sparrow and the house wren. For some species, urban settlements may limit their movement, reducing the connectivity within the metapopulation. This may reduce gene flow, increasing inbreeding and genetic structure among subpopulations. However, for some species, connectivity increases in urban settlements because they can exploit the resources found in urban areas, and we can expect them to experience the opposite genetic effects. Identifying how urbanization affects genetic connectivity of wild animals like birds is important to determine the long-term resilience of these species to urbanization. In this study, we intend to compare the effects of Costa Rican urban areas on population genetics of two bird species with different levels of tolerance to urbanization. This is the case of the white-eared ground sparrow, Melosona leucotis, found in dense thickets, coffee plantations, and secondary forest patches, and Troglodytes adon, house wren, found in open and semi-open areas, forest edges, and previous evidence suggests that this species has benefit from urban development. To study genetic structure in both species, we sampled 5 sites in Costa Rica: Monteverde, a sparsely populated area with coffee plantations and large forest patches connected to mature forest; Heredia, Universidad de Costa Rica, and Jardín Botánico Lancaster, located in the Central Valley of Costa Rica, an area characterized by a high density of populated and urbanized areas, with small secondary forest patches and coffee plantations found within. And finally, Corredores, where we only sampled Troglodytes alone, and this last site is sparsely populated, characterized by intensive oil palm plantations. We captured the birds using mist nets and some playback, and collected blood from 53 individuals of Troglodytes and 56 individuals of Melosone. Then we did the DNA extraction and amplified 8 microsatellite loci on each species. We calculated general population genetic statistics using R, and samples were clustered using structure. We found significant genetic spatial structure in both species, but structure was considerably higher in Melosone than in Troglodytes. Supporting these results, structure software grouped samples of Melosone into two clusters, grouping Monteverde individuals into one cluster and Heredia, UCR, and JBL into a mixture of two clusters. Then, for Troglodytes, the structural analysis cannot accurately cluster individuals, suggesting that all Troglodytes belong to a single large pygmatic population. This contrast suggests a strong difference between the connectivity of both species, Melosone being more restricted and Troglodytes more connected. To test if this difference may be attributed to urbanization, we perform a circuit theory analysis of connectivity using Circuitscape, based on resistance values from land use and cover, assuming that urban areas provide the highest resistance. In Melosone, connectivity, shown in blue, is restricted to coffee plantations, forest areas, and semi-open areas among populations, and resistance increases along urbanized areas. Using this model, structure increases with resistance. For Troglodytes, a similar pattern is observed, suggesting that if urbanization impacts structure, Troglodytes should have a similar structure value as Melosone. However, this contradicts our observations, and Troglodytes has a lower structure. As we will see in the next slide, structure is also affected by environmental climatic conditions. We perform a niche modeling analysis using environmental variables based on the Maxent algorithm, and then we use the habitat suitability values as an input for another Circuitscape analysis. We encounter that Melosone is restricted to the Central Valley of Costa Rica, and its connectivity due to habitat suitability correlates with its structure. However, for Troglodytes, we found it to be distributed countrywide, and its low value of genetic structure among population explains the lack of correlation with climatic connectivity. These results evidence the negative effect of urbanization over the connectivity of one of the species, Melosone. As shown by the animation, during the last decades, its habitat has been lost and replaced by urban areas, especially since coffee plantations and thickets are not included in any of the conservation categories. This could drive the species to a diversity loss, which could ultimately lead to an extinction vortex. Urbanization will have a lasting impact on habitat availability of avian species, and further information on genetic structure will aid the development of conservation strategies. Thank you for listening. Goodbye.
Hello, I am Paula Freitas. I study biodiversity and conservation in University of Brasília, Brazil. My current project is to analyze the urban avian biodiversity, and I want to see, comparing urban and with natural areas, if the biodiversity is significant in urban areas. The difference between natural and urban environments can help us find out more about the adaptation of avian biodiversity in Cerrado, a biome that suffers a lot with human advances. In the left here, we have a map of land use and cover of Federal District, the rectangle in the heart of Brazil, as we can see shown in the map on the right. The black rectangle inside the orange rectangle here on the map on the right. We can see in both green and yellow areas. Green areas are natural ones and yellow are urban ones. On the right map, we can also see the Cerrado area and how the anthropic use of the land affected the biome. So Cerrado is almost all yellow in this map, and that means a lot of urban areas. But how Cerrado is characterized? What is Cerrado? It's the most biodiverse, actually, savanna in the world, and also the second biggest biogeographic region of South America. It occupies 24% of Brazilian territory. is bigger than UK, France, Germany, Finland, and Spain together is surprisingly big. This number is from IBGA, Brazilian Geographic and Statistics Institute. Unfortunately, this biome is highly endangered, and the root of the problem, of course, is politics. Nowadays, Cerrado suffers mainly from agriculture and agropecuarian, but mining also did some critical environmental impact in the past. Only 19% of the biome corresponds today to areas where the original vegetation is still in good condition, and only 0.85% is officially in protected areas. With the exaggerated increase in anthropogenic areas in the biome, the question remains: are birds adapting to the urban environment? What is the difference between the avifauna in the anthropized areas and in Cerrado protected areas? Brasília is the capital of Brazil and is located in the west center of Brazil, in the middle of Cerrado. We have here 4 protected areas in green and 6 anthropic areas in purple. We use McKinnon list over transects and point counts, but how this list works? Well, we use pre-existing trails in the areas and counting 10 species, the first 10 we heard or saw. Here on the right, we can see an example of a McKinnon list. This method focuses on quality of identification rather than speed, so less skilled people can help acquire data calmly and accurately. We process the data with our language in RStudio. The natural areas had higher Simpson diversity, 62.7, and urban sites had 48.9 Simpson diversity. Although species richness is similar between urban and natural point sites, there is a major difference in species compositions when we examine the most common species in each habitat. The Jacquard Similarity Index of the 51 most abundant species was 0.17. Only 9 species in both types of sites, and only 1 species, the Southern Carcará, was in the top 10 in both lists. Here we can see a photo of him. So the sum of the bird species registered list after list is presented in the form of collector curve, or a species accumulator curve. Here in the same site, site, we compared the top 12 species recorded in 2 different methods: McKinnon list, the left, that we use here in this project, and transect on the right, that we use in another project. It has 7 of 12 species in common, 2/3 almost. We conclude that although there may be similar species richness in natural and urban habitat sites, the difference is in dominance, and species composition confirms the need to maintain natural habitats and provides guidelines to manage urban areas to enhance biodiversity. Thank you for listening to me.
Hello, I'm Laura Bizzarri, and I'm here today to talk to you about flower mites that hitchhike on hummingbirds. Imagine a world in which to get food, find a partner, or have children, you have to get on an airplane and fly somewhere else. Now let's substitute human food for tropical hummingbird pollinated plants, substitute human couples for flower mites, and human families for a female mite with her offspring. Lastly, we can switch out our airplane for a hummingbird. The simplified diagram that I just showed you depicts the key players in a system of interactions where hummingbird pollinated plants are also hosts to mites that inhabit their inflorescences, compete with hummingbirds for the nectar of the flowers, and hitchhike on hummingbirds' beaks to disperse to new plants in search of food, mates, and reproductive grounds. I studied these interactions at La Selva Biological Station in Costa Rica, and while studying this system, one of the main questions I had was whether mite dispersal is limited by the hummingbirds they hitchhike on. A combination of field collections, traditional taxonomy, and DNA barcoding were used to identify mites and their interactions with host plants. And to identify hummingbird plant interactions, I used video recordings. We can actually use human transportation systems to understand how accessible different host plants are to mites. I'm from Italy, so let's say I want to go to Rome to get food, then fly to London to find a partner, and finally I want to move to Oslo to raise kids. We have a direct connection between Rome and London, and also between London and Oslo, but there's no direct connection between Rome and Oslo. This animation can be simplified into a diagram with green rectangles for the locations and straight and dashed arrows for direct and indirect access respectively. If we go back to our hummingbird flower mite system, we have this species of flower mite that is found on Heliconia mathiaceae and also has 2 additional host plant species. From Heliconia mathiaceae to reach Amelia patens, the mite will climb on the beak of this hummingbird and hitchhike its way to Amelia patens. Here, in Amelia patens, to reach the 3rd host plant species, the mite will have to climb onto a different hummingbird species and hitchhike its way to Heliconia latispata. So we can see that there is a direct access between Mathiaceae and patens. There is a direct access between Amelia patens and Heliconia latispata. However, there is no direct access or no hummingbird species that visits both Heliconia mathiaceae and latispata. We can once again simplify the animation that I just walked you through into a diagram with each host plant species represented by a green rectangle and lines representing the axis among host plant species. Using a matrix calculations that account for hummingbird visits among host plants, we can calculate a percentage of direct access to host plants out of all of the host plant species a mite uses. After creating the diagrams for all of the 18 species of mites that we find at La Selva Biological Station, we can see that only 3 out of 18 mite species have less than 100% direct access among their host plants. Going back to the main question of whether mites are dispersally limited by hummingbirds, the short and quick answer is no. Instead, mites likely choose their preferred host plants among the options that are offered to them when they're hitchhiking on hummingbirds. Lastly, I want to acknowledge my funding sources and thank my advisor, Dr. Carlos Garcia Robledo, and my field assistant, Mario Alberto Salazar Araya. Thank you.
Hello, everyone. I am Steven Sevillano Rios from Peru. Today, I will tell you how temperature and precipitation can interact to define the species richness in the Andes. Both temperature and precipitation are important factors that define where biodiversity exists on Earth. They are expected to modify the amount of energy available in the systems, and plants and other photosynthetic organisms convert much of the solar energy and water into products, like fruits or seeds that later serve as food to different species like birds. These are byproducts of primary productivity, a proxy of the amount of vegetation in the ecosystem. The Andes are center of high biodiversity. Its high topographic complexity along large latitudinal and altitudinal gradients can strongly modify climatic conditions at local and regional scales, modifying the primary productivity of different areas and producing high alpha and beta diversity. From space, we see that some parts of the Andes are greener than others. Some areas are naturally continuous forests, but in others, continuous forests are uncommon, like in the Peruvian Central Andes. The question is, to what degree are these patterns influenced by climate? Our objective was to examine how temperature and precipitation interact, defining bird diversity in the Andes. We predict that if temperature and precipitation decreases with elevation, species richness would also decrease. If precipitation decreases sharply with elevation, species richness will peak at lower elevation. If precipitation remains constant with elevation, species richness remain relatively constant. Elevation with the greatest precipitation, for example, here at middle elevation, will have the greatest species richness. Elevations with the optimal temperature and precipitation will also have the greatest species richness at middle elevations. We examine 16 elevational gradients, 5 in the northern Andes, 6 in the central Andes, and 4 in the southern Andes. Climatic data was collected over 1,000 points on each of the gradients. Our results showed two main patterns. The first one, where species richness decreased with elevations, specifically in human slopes. And the second, where species richness peak at middle elevation, specifically in the central part of the Andes. When we observe what drives these patterns, we see that temperature is the main driver in human slopes, but the interaction of both temperature and precipitation is most important in drier slopes, here in orange. As we predict, species richness constantly decreases with elevation in most human slopes where both temperature and precipitation also decreases. In drier slopes, however, precipitation increases with the elevation and species richness increased at the middle of the elevation where climatic conditions are neither hotter and drier or wetter but cold. In conclusion, in human slopes, temperature is the main driver of species richness. Here, precipitation is pretty high, so it's not a limiting factor for many of the species. In drier slope, however, temperature and precipitation interact. Here, precipitation is pretty low, lower than 1,000 millimeters per year, so actually acts as a limiting factor for many of these species. Will be important to test these results at global scale. Thank you.
Hello, my name is Adriana Bravo, and on behalf of Santiago Claramont, Millie Wong, and myself, I'll be talking to you about flight efficiency as a predictor of road crossing in Amazonian forest birds. So the ability to move across a landscape is a fundamental property of organisms with implications in ecological processes and patterns, including species distribution, population connectivity, migration, metapopulation dynamics, and foraging strategy. Furthermore, a species' capacity to move should play an important role in determining its persistence in a fragmented landscape and in tracking changing environmental conditions due to climate change. Despite its importance, empirical evidence showing the role of species mobility on patterns of movement across fragmented landscapes is still limited. In this study, we determined the role of flight capacity on the ability of tropical birds to cross a man-made gap in Amazonian terra firme forests. For that, we used data on recaptures of banded birds from a study by Lawrence et al. In an area of terra firme forest divided by a road in the Brazilian Amazon. In this study, the researchers recorded whether recapture occurred on the same side of the forest where the bird was banded or across the road. With this information, for each species, we estimated a probability of road crossing. To estimate the flight capacity of each species, we calculated the hand-wing index, which is a proxy for the aspect ratio of the wing, and for that reason is related to flight efficiency. We obtained all measurements from museum specimens. In addition, we collected data for these 3 other ecological predictors to be assessed in the study. Diet, so we classified the species as insectivores or omnivores. Flocking behavior, we determined whether a species were part of a mixed species flocks or not. And foraging stratum, with ground, understory, mid-story, canopy as the different levels of foraging. Then we analyzed the relationship between these 4 ecological predictors and the probability of road crossing by using phylogenetic logistic regression models. So we analyzed a total of 45 species. The handwind index among these species ranged from the lowest value of 6.6 for Microcirculus bambla to the highest value of 30 for Geotrigon montana. We classified 35 species as insectivores and 10 as omnivores. 30 of the species were classified as non-flocking species, while 15 were classified as flocking species. So as you can see in these figures, only the handwind index has a significant relationship with the probability of road crossing. Each dot is a species and the size of the circle indicates the total number of recoveries. So here you have a table that shows the multi-predictor models. The best multi-predictor model included the handwind index, diet, and flocking behavior. However, this model was followed by several other secondary models of similar fit, resulting in considerable model uncertainty. Note that the handwind index is present in all best models while other variables are inconsistent. So when we calculated variable importance based on model probability, only the handwind index stands out as an important explanatory variable. So in conclusion, we can say that flight capacity in birds has a strong influence on the probability of road crossing among Amazonian forest birds, and that the contribution of other ecological factors is ambiguous. Therefore, the handwind index can be used to rapidly assess the vulnerability of a species in fragmented landscapes. So with that, I want to finish by acknowledging the institutions that support this project. Thank you very much.
Hello, my name is Luisa Garra de Souza and I'm an undergraduate student in biological sciences at University of Brasilia, and I'm here to present a research using automatic sound recording to monitor avian diversity and guide conservation in an urban natural landscape in a biodiversity hotspot. Many tropical cities are situated in global biodiversity hotspots, and their growing human population and land use are negatively impacting endemic and/or threatened native species and communities. The city of Brasilia, Brazil, is surrounded by Cerrado, the most species-rich tropical savanna on the planet, of which over 50% of its original extent has been converted to a human use in recent decades. The purpose of this project is to use automatic recording of bird sounds to generate biotic diversity indices for various urban and natural environments, and to use these indices to direct landscape restoration and management to increase diversity and representativeness of Cerrado fragments in urban parks. We placed cost-effective audio move recorded from April 2019 to May 2021 in 12 ponds in 3 protected areas, 4 ponds in 1 urban ecological park, and 7 ponds in urban residential areas and gardens. We used RStudio to analyze the data extraction from AudioMove. When we analyzed the data, it was noted that the natural and semi-natural areas profiles are very similar. Difference between ponds is more related to the seats than to the time of day. The analysis showed that there are significant variance between natural and urban areas. When we look at the me and site graphic, we see no clear patterns between sites, just as the graphic me and die. However, the natural areas are more different than the anthropomorphized areas. When you look at the graphic, mean Eve appears to have a tendency for more natural areas to present a lower rate. And when you look at mean Bio graphic, shows that SKN 314 moved a lot from other seats, which would indicate that it's an area with a lot of cars, for example. As we can see, the graphic MIA-H and MIA-M separate well urbanized areas from natural and semi-natural areas, and the graphic MIA-C show that biophony rates are higher in more natural environments. This graphic PCA-centered correlation for all sites by time shows us there is no big difference between sites during times of a day. And the PCA-centered correlation for all sites graphic demonstrates the more urbanized area shows greater variation in relation to natural and semi-natural areas. So, what you can conclude, natural areas and semi-urbanized areas have a similar profile to each other. The PCA analysis also shows that semi-urbanized areas have a similar profile to each other, and urbanized areas shows a low index deviation from the other indexes. And here, for more information, we have some acronyms and local names we used in our research. We would like to thank to LabVIEW Professor Ricardo Machado, to Parque Nacional ICMBio, AudioMove, RStudio, UNB Secretaria de Meio Ambiente. Also, thanks to Fazenda Água Limpa and Reserva Ecológica from BGE for support the project and allow us to use the area to install recorders. Thank you all.
Well, great. Congratulations to all the speakers. Those were really wonderful talks. How about a round of applause for everyone? You can, even though you may not be able to hear it, you can, we are applauding everyone. Thank you so much. So as a reminder, if you have any questions, please put them into the Q&A or put them into the chat, and I will read them out. We have one question. Adriana posed one to VTech, and she was asking on your study about, for your study, would the preference by birds for darker and cooler places in the understory be related to a preference for the insects that they feed on?
Yes, thank you for the question. I think that's certainly a possibility. The problem with my study, we just know the average conditions the birds experience, and the question is why they are experiencing those conditions. Are they in certain places because they feed there on insects, which I think what Adriana is suggesting, and I think that's a great hypothesis. I think there's a lot of evidence that microclimate is highly influencing the invertebrate communities, especially in the forest floor litter where these birds are feeding. So I think that's certainly a possibility. Yeah, I just couldn't tell why birds were in certain places. Especially these guys are highly secretive and they're almost impossible to see. So we don't really know what they were doing in those places. I think it's a good question.
Great, thank you. And Otieno, you had a comment in the chat as well. Would you like to add to that? Okay.
Just in case Otieno is not plugged in here,
Okay.
I can just try to elaborate on that. I think invertebrates are ectothermic, so they might be particularly affected by things like temperature and humidity. So I think the probability that it is because of these invertebrates are being influenced by something is high. And Otina can chime in when he wants to.
We can hear you.
Oh yeah, thank you.
Yes.
Oh yeah, my thought was that if the birds are retreating the hidden and cooler areas, it's most likely that they depend on forest floor invertebrates, especially those ones that are detritivores, which they depend on and supports their tendency to go reclusive, so they can hide, as he says. They are basically birds that hide and are difficult to see and difficult to find, and they find it more easier to forage very close to the floor where they can pick the insects underneath the forest litter, as opposed to flying high up in more exposed areas where they can easily be spotted. So the detritivores invertebrates are likely the most important component of their diets, which supports their microhabitat preferences. That's what I thought. I don't know if that answers some, contribute to it. Is it likely that there were species that depend on small invertebrates on the forest floor, and that those places where they were hiding are most likely the coolest areas close to the forest litter down on the floor, as opposed to up in the upper story? That's just what I thought.
Okay. And then I think there's a question to Laura about the mite species. So do the mites use the different plant species for reproduction or for feeding before reproduction?
So they can use any of their host plant species to do any of their activities. So as far as I know, there's no evidence that they prefer one host plant species for reproduction and another one for feeding. So yeah.
So whichever one is fine for them?
As long as it's, yeah, one of their preferred host plant species that they use, they can do any of their activities on them.
So there hasn't been any, we don't know that there's difference in fitness on different hosts. It's just,
yeah.
Okay, great, thank you. I had a question for Akshay. I was wondering, in the system of fragments that that you've been studying, when I missed it of when they were fragmented. So was it right before the study, or yeah?
No, so these are we assume that these are in steady state. So they likely they were fragmented before independence, so at least 70 years ago, and the records aren't clear. So maybe in the turn of the century, about maybe 100 years ago. And the thing is, so they were initially coffee, and then that became tea. So in some sense, it became a soft edge to a hard edge. But tea has persisted for the last 70 years, the matrix, and the forest fragments are embedded in there. And the boundaries have remained stable as well.
Okay, so that seems pretty amazing to me that they've been fragmented for that long, and then just over your 6-year period, they declined in density by 30%. That seems pretty dramatic.
Yeah, definitely. We think that these are probably being driven by regional trends. So there was a recent study called the State of the India's Birds that used eBird data to draw population profiles of birds in the country, and this is a persistent pattern across the southern Western Ghats of India and in many parts of India as well. So we think that might be a reason, but we're also cautious because there were 3 different observers at different points in time, which was inevitable. So we also think that that could have contributed to some of the trends.
Okay, great, thank you. All right, Stephen has a question for Supun and Luisa about, I think Luisa is not on right now, but what type of considerations should we take if we want to conduct a long-term acoustic monitoring program in, for example, a national park, so that they can help to improve the management of the area? That's a great question. So if we're going to do a long-term monitoring program, how can we best do it using acoustics? Yeah.
Yeah. Hi. Sorry, I'm in a very noisy area actually, so I don't know if you can hear me really well, because I can't hear you really well actually. But yeah, in short, I would say, but I tried to during my study was to understand if we can use this acoustic indices, means one type of acoustic monitoring method in these tropical ecosystems and subtropical ecosystems, which included both forest and agricultural areas. So to answer that question, I would say acoustic indices is a really good method. But what I wanted to highlight there in my study was also we need to develop a lot regarding these tropical ecosystems if we consider tropical forests. But when it comes to temperate forests, it has been used as a really good, not really good, but yes, a considerably useful technique to understand the biodiversity. And so for that, actually, we can use long-term monitoring processes using acoustic devices, different kinds. We can put song meters. We can fix the recorders for like years, and we can capture a lot of recordings, a lot of acoustic data. And actually, we can it's not like using point counts or like conventional methods, because it's like we can collect a lot of data using these acoustic recorders. So such kind of methods are really actually useful especially in temperate regions. But when it comes to tropical areas, like as the areas we considered in our study, we actually need to improve a lot, because there is a lot of actually disturbances in the natural ecosystem itself. Like, let's say insect vocalizations, and like compared to temperate regions, in tropical regions, we have a lot of insect calls. If we want to monitor birds and if we ignore the insect vocalizations, then actually methods like acoustic indices actually are not giving really good results actually. So yeah, so when it comes to tropics, we need to develop these methods a lot more. And we need to consider these disturbances when we calculate these indices. That's our main conclusion was. So yeah, I would say acoustic indices is a really good method if we ask the short answer for that question. But yes, we need to improve a lot. Thank you for the question. I don't know if I actually gave at least a little small answer. I don't know. But yeah, thank you.
Great, thank you.
You're welcome.
So we have about 5 more minutes. If you have a question, please feel free to add it to the chat. I'll ask one while we're to give people a chance to put put anything else that they would like to know. Mauricio, I was wondering, so you compared these two different species and you found pretty different, pretty big differences in their genetic structure. Do you think that they can be, or what kinds of information can we use to generalize from them? Like, would we be able to say that, you know, X number of species would be like the Melazoni versus the Troglodytes?
Well, we can say that the connectivity, well, thank you for your question. The connectivity between the patches in Melazoni is really low, okay? And we can extrapolate this information to other birds who inhabit dense thickets in Costa Rica. This is because this area has been reduced. It has been fragmented a lot during recent years, during 30 years, and also coffee plantations. So we can say that probably species who inhabit dense thickets in these areas might be experiencing a higher genetic structure as well. So we can, and one of the projects that there is currently in Costa Rica is to improve the management of these areas because they are not included in any management or any conservation program. So that is the idea, to further increase conservation in these areas and in these habitats to prevent from local extinction.
Great, thank you.
You're welcome.
And I have another, oh, here we go. So VTech, would you, Akshay has a question about, he was curious about the dip in body temperature at 6 p.m. for a lot of the birds. Do you have an explanation for that?
It's a good observation. Yeah, that struck us as well. And the best what we can say right now that it's related to their bathing schedule. There is some evidence from other parts of Amazonia that these tropical forest birds can have very consistent bathing schedule. Just seems like right before they go to bed, they must dip into these streams or some kind of water bodies that are nearby. That results in this kind of big drop in temperature, about 3 degrees sometimes Celsius. So that's our interpretation. They must be bathing, and that pattern is evident, especially in some species like those leaf tossers, the scleru species. But most of them have some kind of a dip right before it gets dark. And we think it's bathing.
Huh. That's pretty fascinating. The bird baths causing that. And in our last minute question for Laura, is there rare indirect dispersal that mites can take advantage of? So do they have to go on the hummingbirds?
So in the example that I showed in the video, like the mite has 3 different host plants, but it can only directly access 2 of them. And so like to get to the 3rd host plant species that it uses, it actually has to sort of get to like the intermediate host plant. So you can imagine it'd be like if to get to a certain city by flying, you have to stop and you have like a layover. So they do have sometimes indirect dispersals, but they always use hummingbirds to do that.
Okay, great. Well, thank you again to all of our speakers today. Really great lightning talks. We have about a 10-minute break now between now and the next session. So I hope to see you all in one of the later lightning talks or panel sessions. Have a great 10-minute break here, and we'll see you all back in about 10 minutes at the next link. So thank you, everyone. Congratulations. See you soon.
Thank you.
Thank you very much.
Thank you.
Bye.
Thank you.
Bye.
Thank you.